Bacterial conjugation is a process in which mating clusters of gram-negative and positive bacteria, such as E. coli, transfer genetic material in the form of fertility plasmids via cell-cell contact. The cell containing the F plasmid is known as the donor. The cell that receives the F plasmid is known as the recipient. These fertility plasmids hold a series of genes known as the TRA group. Half of these genes code for the mating pair formation system, and the other half codes for the DNA transfer and replication system. The MPF system includes pili that attach to the recipient cell. These pili then retract to form the mating bridge. A DTR enzyme called relaxase breaks the sugar phosphate backbone of the plasmid at the origin of transfer. Relaxase then binds to the end of the loose strand by replacing the last organic base with its own tryptophan. DNA helicase can now separate the two strands by breaking the hydrogen bonds between them. Relaxase is now essentially a guide for the free strand. The secretion system shoots the relaxase through the membrane of the two cells, and the attached DNA is pumped through by DTR coupling proteins. Another DTR enzyme called primase accompanies the strand into the recipient cell. Its role is to create primers. The relaxase then recircularizes the strand. The newly created primers can now adhere to the strand. DNA polymerase can now use free nucleotides to create a second strand, completing the plasmid. The same process is occurring in the donor cell simultaneously. Once the two plasmids are complete, the cells separate. The recipient cell, now known as transconjugate, is capable of forming its own pilus and transforming other cells.